Hi, thank you so much for the introduction. It's so nice to be here with you all today. Thank you so much to the organizers for having us here. I'm Rebecca James, as Emily mentioned, and today I'd like to talk to you about the evolution of our therapeutic target, HDAC6. So, just a disclosure, I am an employee of Eikonizo Therapeutics, as I mentioned. At Eikonizo, we are working on developing highly selective, orally available and CNS-penetrant HDAC6 inhibitors for neurodegenerative diseases. But what is HDAC6, and why should you care about it? Sorry, one second. I'd like to spend a little bit of time with you today sharing the rationale underlying our interest in targeting HDAC6 for neurodegeneration. I'll talk about some of the implications of HDAC6 in neurodegenerative diseases, and then I'll discuss how we think HDAC6 inhibition can ameliorate the pathobiology of neurodegenerative diseases, and then I'll share some of our preclinical data supporting our selective HDAC6 inhibitors for ALS, frontotemporal dementia, and also Parkinson's disease. And finally, I'll touch base briefly on our clinical development plans. Okay. So HDAC6 is a protein with two catalytic deacetylase domains, CD1 and CD2, which remove a chemical group called an acetyl group from its substrate proteins, not unlike a pair of scissors cutting off a string from your favorite sweater. It also has a C-terminal ubiquitin binding domain, and as the name suggests, it's one of several HDACs, 18 in total. Unlike most HDACs, which remove acetyl groups from histones in the nucleus to turn off gene transcription, HDAC6 has two nuclear export signals and also an SE14 domain that promote its cytoplasmic retention. And that allows it to interact with substrates in the cytoplasm and deacetylate those substrates, which are relevant for intracellular transport, like its primary target, alpha-tubulin, and also allows it to interact with ubiquitinated proteins that are aggregated or damaged organelles to promote their clearance via autophagy. Okay. And these two activities are critical for the health of neurons, which require effective cargo transport along their microtubule highways to bridge the very long distance between their cell bodies and their terminal targets. Okay, and when you think about this in the context of ALS and spinal motor neurons, that distance can be up to 1 meter long, considering motor neurons in the spinal cord and then targets all the way out in the foot, for example. In healthy neurons, HDAC6 contributes to the, sorry, I'm feeling a little nervous. In healthy neurons, HDAC6 contributes to the optimal balance of tubulin acetylation, which facilitates microtubule stability and motor protein binding, which subsequently enables protein and organelle clearance and quality control. So if HDAC6 helps to keep neurons healthy, you may be wondering why we're interested in targeting it in neurodegenerative disease. HDAC6 was first cloned in 1999 and characterized as an alpha-tubulin deacetylase shortly thereafter. In 2003, a study from Xiaopeng Yao's lab at Duke came out that first implicated HDAC6 in neurodegenerative diseases when it was found to co-localize with alpha-synuclein in Parkinson's disease, and also in dementia with Lewy bodies. Since then, a great breadth and depth of literature that has been published implicating HDAC6 in various neurodegenerative disorders. Now, don't worry, I won't bore you to death by reading this laundry list of studies, which is not nearly exhaustive. But what I hope you can take away from this is that over 20 years of literature points toward roles for HDAC6 in CNS-relevant biology and in neurodegenerative diseases. In particular, I've bolded studies demonstrating how HDAC6 inhibition of that catalytic deacetylase activity is neuroprotective. So what are the implications for HDAC6 in neurodegenerative diseases, and why would its inhibition be beneficial? In neurodegenerative diseases like ALS, misfolded proteins aggregate and can directly impair axonal transport by blocking the microtubule highways. Misfolded proteins can also enter into and damage mitochondria, leading to oxidative stress and reactive oxygen species production, and the reactive oxygen species then further damage proteins, and all of this can feed into and cause neuroinflammation. And then neuroinflammation can further cause oxidative stress within the neurons, causing this vicious cycle that ultimately underlies neuronal demise. Interestingly, in several neurodegenerative diseases, HDAC6 levels have been shown to be increased, and HDAC6-mediated hypoacetylation can destabilize transport, further impairing proteostasis defects and other cellular functions and contributing to that vicious cycle. We believe that through selective inhibition of HDAC6, we can stabilize intracellular transport and preserve the beneficial roles of HDAC6 in proteostasis, and that together, by combining these net effects of improved intracellular transport and improved proteostasis, we can ultimately improve neuronal health and function. Okay. And we think that this is particularly important in ALS, for example, because, as I mentioned, the spinal motor neurons need to transport their cargoes a very long distance, which makes them more susceptible to transport instability. Furthermore, there's been several genetic mutations identified in transport machinery in ALS that affect the transport of multiple cargoes, including proteins, mitochondria, and mRNA, and that's seen in both sporadic and familial ALS. The same is true for many genes encoding autophagy machinery, and as you're all well aware, there's TDP43 pathology in more than 95% of sporadic ALS. There's also aggregates of SOD1, FUS, and tau. That's observed in both sporadic and familial ALS. And we think that these defects in intracellular transport and proteostasis in ALS in particular lead to impaired neuronal health and function. And our therapeutic hypothesis is that by correcting these axonal transport and proteostasis defects, we can bring benefit to all persons with ALS. So I hope by now you agree that ALS is a target of interest for neurodegeneration broadly and for ALS specifically. So if so, you may be wondering why an HDAC6-related therapeutic option hasn't become available by now. Small molecule HDAC6 inhibitors have actually been in development for several years for several indications. However, the first generation inhibitors had many limitations that precluded their use as therapeutics. The first gen HDAC6 inhibitors had poor pharmacokinetic and safety profiles. These were hydroxamic acid scaffold inhibitors. Our HDAC6 inhibitors, on the other hand, have an oxidiazole scaffold, which is highly potent and has a favorable pharmacokinetic profile, allowing for once daily oral dosing. The first gen inhibitors also had poor selectivity and off-target effects, whereas our HDAC6 inhibitors are highly selective for HDAC6 relative to all other HDACs, leading to fewer off-target safety concerns and a better therapeutic index. The first gen inhibitors also had poor CNS penetration, whereas our HDAC6 inhibitors are highly CNS penetrant, which uniquely enables Eikonizo to address neurodegenerative diseases. And sorry, the animation has gone a little off here. Additionally, the first generation clinical trials didn't have predictive biomarkers, so clinical efficacy was poorly predicted. However, we have a clear plan focused on target engagement and mechanistic biomarkers to facilitate and to improve dose and patient selection in the clinic. And to exemplify the superior selectivity of our compounds, what you can see over here is that our lead development candidate, EKZ-102, is highly selective for HDAC6 relative to all other HDACs, whereas ricolinostat is not very selective. It was only 7-fold selective and failed in development, whereas 102 is more than 1,000-fold selective for HDAC6. And here's some exemplary data from a mouse pharmacokinetics study showing high exposures of EKZ-102 in the brain and spinal cord that are well above the biochemical IC50 for HDAC6, supporting the application of 102 for neurodegenerative diseases. We've characterized both oxidizole tool compounds and EKZ-102 in a battery of in vitro and in vivo studies. And we've seen robust preclinical efficacy across multiple models. Given our time constraints, though, I'll share just a subset of our preclinical data package, highlighting some studies from our recent manuscript that was published online in Brain just last week. Okay, so first off, in a primary rat motor neuron glutamate excitotoxicity culture, which is a model of SOD1 familial ALS. I'm sorry, it's not advancing. Okay. In this model, we treat the spinal motor neurons with an excitotoxic concentration of glutamate that induces a robust accumulation of misfolded SOD1 in the cytoplasm. And what we can see is that our HDAC6 inhibitor strongly suppresses that, outperforming a pharmacologically relevant concentration of riluzole. We've also measured oxidative stress in this system using a mitochondrial reactive oxygen species probe, MitoSox. And our HDAC6 inhibitor strongly suppresses reactive oxygen species. And by combining the benefit of improving both of these compromised pathways, we see increased neuroprotection. So here we're showing an improvement or protection of the neurite network with HDAC6 inhibition and a very robust neuroprotective effect, as we could see increased neuronal survival. Okay. We were, however, unable to evaluate our mechanistic pillar of axonal transport in this system, so we next turned to an imaging-based technique to quantify net retrograde axonal transport in SOD1 mice using a recently published elegant technique developed in the lab of Dr. Bob Brown. Okay. In this system, so we were very fortunate to collaborate with Dr. Brown and his lab for this study. We worked very closely with Dr. Justin Lee, who developed this radio-labeled tetanus toxin C fragment that is injected into the gastrocnemius muscle of the mouse, and then it's retrogradely transported into the spinal cord, and then Justin imaged the amount of signal that was taken up into the spinal cord over a 2-week period of time by SPECT imaging, or single photon emission computed tomography. And what you can see is that over the 2-week period of time, there's a significant increase in the amount of signal that accumulates in the spinal cord with HDAC6 inhibitor treatment. And importantly, these improvements in axonal transport were associated with an improved motor function. When we measured the grip strength of these animals over time, you could see a significant increase with HDAC6 inhibitor treatment relative to the vehicle treatment group. And then we saw the same thing when we measured rotarod performance. Next, since we recognize that there's no true gold standard ALS animal model that predicts clinical benefit, a clinical survival benefit, we prioritized a biomarker-focused study with ALS TDI so that we could identify and preclinically validate HDAC6 inhibition responsive biomarker candidates for consideration in our clinical trial design. We treated SOD1 and wild-type mice with vehicle and a group of SOD1 animals with HDAC6 inhibitors from postnatal day 42 to postnatal day 105 when the mice are symptomatic. And here I'm demonstrating a heat map of biomarkers that was identified from a NULISA-seq experiment where we evaluated the levels of different CNS-related biomarkers in terminal plasma. And we saw a strong conversion of the inflammatory biomarker signature of the SOD1 mice back to a more wild-type signature. Just as an example, if you look at CXCL13, it's blue in the wild-type mice. It goes up to red in the SOD1 mice, so the expression is increased, and then it drops back down to blue with HDAC6 inhibitor treatment. So we saw a conversion of the signal. Okay. And given its prognostic importance in neurodegenerative diseases, we assessed NFL in terminal plasma independently and detected a significant reduction in plasma NFL with 2 of our HDAC6 inhibitors, supporting a decrease in axonal degeneration. And we also saw an increase in the muscle mass in those two HDAC6 inhibitor treatment groups, which is indicating that there is, or supporting that there's neuromuscular junction preservation with HDAC6 inhibition, because muscles will atrophy in response to neuromuscular junction denervation. Okay. And together, these benefits were associated with improved motor function for both HDAC6 inhibitor treatment groups, as we saw that there were no animals in the HDAC6 inhibitor treatment groups with a neuroscore greater than or equal to 2, which indicates the onset of paresis in the hind limbs, whereas in the SOD1 vehicle group, there were 27% of the animals that had a neuroscore greater than or equal to 2. We also evaluated efficacy of our compounds in models relevant to both sporadic ALS and FTD. Here we're looking at human cortical-like eye neurons with a TDP43 M337V mutation, and we measured the fraction of moving mitochondria in this system in collaboration with Dr. Sami Barmada. And what you can see is that there's a nice dose-dependent increase in axonal transport with HDAC6 inhibition. And we also looked at proteostasis by measuring the accumulation of TDP43 in the cytoplasm of TDP43 M337V motor neurons. These are, again, human motor neurons. And we saw a very strong suppression of cytoplasmic TDP43 accumulation. Additionally, when we looked in these neurons at Stathman2 cryptic exon mRNA, we could see a significant reduction with HDAC6 inhibitor treatment, suggesting that we're improving nuclear TDP43 function. And then we also looked in vivo at phospho-TDP43 aggregation in the TDP43 delta NLS model developed by Adam Walker. And we could see a significant reduction in TDP43 pathology, and also a reduction in the number of GFAP-positive astrocytes, indicating that we're lowering in neuroinflammation. So if our therapeutic hypothesis is correct, then we should expect to see improved survival by combining the protective effects of these pathways, which we have seen in multiple systems. But in the interest of time, I just wanted to show you the data from our human system here. These are studies, longitudinal fluorescence imaging of survival that was performed in collaboration with Dr. Sami Barmada. In this system, we're looking at the survival of cortical-like eye neurons. These are bearing the TDP43 M337V mutation. You could see that in red, there's an increased cumulative risk of death relative to the isogenic control in black. And our HDAC6 inhibitor treatment completely restored the survival of the mutant neurons back to control levels. We also looked at survival of motor neurons. These motor neurons had the G298S mutation. You could see that there's an increased cumulative risk of death that's strongly suppressed by HDAC6 inhibitor treatment again. Furthermore, we've looked at intracellular transport in models relevant to C9orf-related disease. Here we're using healthy human motor neurons that are injured with a C9orf72 dipeptide repeat, which impairs axonal transport. And you can see that our HDAC6 inhibitor treatment strongly increases axonal transport in the system of mitochondria. And when we looked at survival in C9orf72 mutant cortical-like eye neurons, again, we saw that our HDAC6 inhibitors strongly decrease the risk of death of these neurons. We additionally recently looked at C9orf72 mutant motor neurons as well, and again saw a significant reduction in cell death. Okay. In addition to these robust preclinical data in models of ALS and FTD, we're seeing similar protective effects in models of Parkinson's disease. As you can see here, this is a system where we're treating dopaminergic neurons, primary rat dopaminergic neurons, with alpha-synuclein preformed fibrils, and that induces alpha-synuclein pathology, and our HDAC6 inhibitor strongly suppresses this. We also saw similar reductions in mitochondrial reactive oxygen species. and reductions in reactive microgliosis, as we saw a decrease in the number of IBO1 positive microglia. And again, by combining the net protective effect of these pathways, we could see an improvement in neuronal survival of dopaminergic neurons. We were very fortunate to recently receive a phase 1 SBIR award to advance this work, so stay tuned for more preclinical support of our lead development candidate, EKZ-102, in neurodegenerative diseases. I hope that cumulatively these results and our passion for HDAC6 inhibition have piqued your interest in this target. So to summarize, I've shown you so far that our HDAC6 inhibitors improve intracellular transport in models relevant to ALS and FTD. They improve proteostasis of several pathological protein species, including SOD1, TDP43, and alpha-synuclein, which is important because alpha-synuclein can seed TDP43 and SOD1 pathology. We've shown that we can reduce oxidative stress in cellular models of ALS and FTD and reduce neuroinflammation, and that together, these effects lead to increased neuroprotection and improved function. So to translate these potential therapeutic benefits into the clinic, we need to establish a reliable method to assess target engagement in order to improve dose and patient selection. Since alpha-tubulin is a primary substrate of HDAC6, we've been developing methods to evaluate alpha-tubulin acetylation. We've looked at the relationship between HDAC6 inhibitor pharmacokinetics and target engagement in SOD1 mice as part of our preclinical efforts. And as you can see, there's a really nice correlation between exposures on the left and target engagement, demonstrating an excellent relationship between compound exposure and target engagement. And to evaluate target engagement in the clinic, we've been developing a flow cytometry method to assess acetylated alpha-tubulin in isolated PBMCs. On the left, I'm showing you in vitro proof of concept for measuring HDAC6 target engagement by flow cytometry. Our selective HDAC6 inhibitors increase tubulin acetylation here in blue, but they do not increase the acetylation of histones, which you can see on the right, indicating and supporting their specificity. We're currently adapting this protocol for the clinic, where we'll focus on target engagement and also biomarkers of disease activity, which you could see here. In our, sorry, I didn't mean to do that. In our phase 1 studies, we will measure acetylated tubulin in PBMCs to verify target engagement. And because HDAC6 inhibition has multiple beneficial pathway effects, we'll also be looking across a wide range of relevant disease activity biomarkers. Here's just a few examples. Our phase 1A will be in healthy volunteers so that we can fine-tune our target engagement biomarker to refine dose selection. And for our clinical plans, EKZ-102 is currently in IND-enabling development. We're hoping to file a IND or CTA in 2026. And as I mentioned, our phase 1A will be in healthy volunteers, and then we hope to extend that to a phase 1B to extend our target engagement data set and also to pilot our biomarkers. We are extremely excited by the promise of our lead development candidate, EKZ-102. And we hope that this will offer a disease-modifying approach for sporadic and familial ALS. So with that, I'd like to thank you for your attention and take questions.
Thank you, Rebecca. I think we have time for one question, if we have any from the audience. Do we have any online, Megan?
The question is, how do you anticipate achieving selectivity given HDAC6 multiple roles, including in autophagy?
Including in autophagy?
Yes.
Okay. That's a really interesting question because, HDAC6 deacetylase activity is important. And I think like a lot of drug targets, it's going to be important to titrate the right amount of any therapeutic. So that's why we are really focused right now on targeting and identifying and defining the right amount of target engagement that's going to achieve the effects that we hope to see. So our goal right now is just first to truly define how much target engagement you need to then prevent other downstream consequences that you might not want to see.
Awesome. Thank you, Rebecca.